welcome to the deep dive. Today we're really diving into something crucial, the absolute cutting edge of Alzheimer's disease research and treatment.
That's right. We're drawing insights from a really fascinating panel discussion featuring some, top experts in the field.
Exactly. We're trying to understand where we actually stand in this fight. And by hearing from these people on the front lines, well, the idea is to pull out the critical knowledge and give you a clearer picture. The challenges, the potential breakthroughs.
Yeah. We'll be hearing from Jeff Cummings, University of Nevada, Las Vegas, Howard Fillit from the Alzheimer's Drug Discovery Foundation, or ADDF, Miia Kivipelto, Karolinska Institute.
Big names. Reisa Sperling, too, from Harvard and Mass General.
Right, and Philip Scheltens, who's moved into venture capital with EQT Life Sciences, and Geoff Kerchner from Roche, handling their late-stage trials.
Quite a lineup. So our mission here, for you listening, is to extract the really vital stuff about drug development, prevention strategies, the role biomarkers are playing now, which is huge, and just the overall direction things are heading.
We want to make this complex area a bit clearer, focus on what's getting prioritized, the hurdles, and, what breakthroughs might be just over the horizon.
Okay, so let's kick off where a lot of this starts, or maybe stalls, getting the funding. Philip Scheltens, the venture capital guy, he had some pretty eye-opening things to say about how those decisions get made.
Yeah, incredibly high stakes.
Absolutely. Okay, let's unpack this. Philip mentioned getting something like 400 proposals, slide decks, he called them, every year.
That's right, about 400. And the really striking part, they only fund about 1% of those.
1%. Well, that tells you something about how intense the competition is.
It really does. It means only the most rigorously scientific, the most well-supported ideas even get a look in. It just highlights the sheer difficulty of tackling Alzheimer's.
So what filters do they use? How do they even start sorting through that pile?
Well, Philip laid out some key principles. First off, the target for the drug has to be biologically valid. There needs to be solid science behind why you'd go after that particular thing in the brain.
Right. Makes sense. A scientific reason.
And then, ideally, it should be genetically validated. So evidence from human genetics linking that target to the disease itself.
Okay. Following the clues our own biology gives us, is that genetic validation almost like a must-have now for getting serious funding?
It's certainly a very, very strong plus. Then you absolutely need biomarkers.
Biomarkers, right? The measurable sign.
Exactly. Indicators that tell you if the drug is actually hitting its target in the body. Without that, you're kind of flying blind, aren't you?
Yeah. You wouldn't know if it's doing what you think it's doing.
And then, of course, solid proof of concept in preclinical studies. You made good lab data, good animal model data before you go into humans.
And the team running the show matters too, I assume.
Oh, definitely. Philip emphasized that. The quality, the track record of the company's team, heavily scrutinized. You need people who know what they're doing.
Makes total sense.
And interestingly, he stressed that their decisions are really driven by the field. They're not picking targets in advance. They're reacting to the best science that's emerging.
So staying flexible, following the most promising leads as they pop up.
Yeah.
Okay. Now let's switch gears a little. Howard Fillit from the ADDF, he brought up something else critical, the link between academia and industry.
Yeah, that collaboration. It sounds like it's changed a lot.
It really has. Howard pointed out that maybe, say, 20 years ago, seeing drug development start in a university lab was, well, pretty unusual.
Right. But now, he says it's common, almost essential. These partnerships between universities, biotechs, big pharma, they're the norm.
That shift feels really significant, doesn't it? So what role does the ADDF play in all this? Are they just funding or more involved?
Oh, much more involved. Howard explained they have, I think, 14 neuroscientists on staff. They're actively guiding the researchers, offering expertise, helping them navigate that really tricky path of drug development. They're not just writing checks.
Wow. And the scale is huge, right?
It is. He mentioned funding over 700 programs, 20 countries, something like 150 biotechs over the last couple of decades. That gives them this incredibly broad view of everything happening.
And they use this term, venture philanthropy. What exactly does that mean?
It's a really interesting model. Because they're a nonprofit, Howard explained, they can tolerate more risk than traditional VCs. And they can stick with projects for longer.
So they're playing the long game, even if it takes, say, 20 years for something to potentially pay off.
Exactly. That's a serious commitment to the science.
Yeah, it really is.
And their funding approach is kind of a hybrid. They vet the science like a grant agency would, very rigorously, but the funding structure can look more like a venture investment. The key is focusing the money directly on executing the scientific plan.
Okay, got it. So moving from funding and structure to prevention, Reisa Sperling was talking about biomarkers in that context. There's been so much focus on clearing amyloid. Are we getting to a point where just changing a biomarker could be enough for approval? For prevention, I mean.
Reisa thinks we're not quite there yet, but the hope is definitely there. She stressed that we really need trials that show a clear link, a definite connection, between changing a biomarker and actually delaying cognitive decline in a way that matters to people.
So seeing amyloid go down on a scan isn't enough on its own. You have to prove it helps people think and remember better in their lives.
Precisely. But she is optimistic. She thinks some of the upcoming secondary prevention trials might provide that evidence.
Secondary prevention meaning people who have the biomarkers but no symptoms yet.
That's right. And she also hopes these trials will look beyond just amyloid and tau, bring in biomarkers for things like synaptic health.
Synaptic health, the connections between the brain cells.
Exactly. Getting a more complete view of brain health, not just the pathology. And maybe, just maybe, that could help shorten how long these trials need to run to see an effect.
Makes sense. What about that cognitive test sheet developed, the PAS-CC5? Can showing a difference on that be enough for regulators?
Well, she hopes so. The significant difference between the drug and placebo on the PAS-CC5 she thinks could potentially be enough. But she was quick to add, it depends. Ultimately, it's the regulator's call.
Right, but her point stands, cognition is functionally meaningful, especially early on. Even small changes matter then.
They absolutely do. It affects independence, quality of life. But she also brought up a tricky problem. These phase 2A trials where you do see biomarker changes, but the clinical results aren't quite strong enough.
The biomarker positive, clinical outcome negative issue. That must make it hard to get more funding for the next phase.
Exactly. It's a real hurdle. And it underlines why we need these big, long trials right now to really nail down the link between biomarkers and clinical outcomes.
So we can eventually use biomarkers to manage Alzheimer's like we manage, say, cholesterol for heart disease.
That's the ultimate goal Reisa pointed towards, using biomarkers for chronic disease management, just like in other fields of medicine.
Okay, let's bring in Geoff Kerchner from Roche. He's dealing with the early stages, deciding which drugs actually move forward. With limited resources, they must have to be pretty selective.
Picky was the word he used, very picky. And he stressed how important it is to, kill off the unpromising candidates early. Don't keep pouring money into something that's not working.
Makes sense financially and ethically too, probably.
And he sees phase 2 trials as the absolutely critical step, but also often the most flawed part of the whole process.
Interesting. Did he give examples?
He did. 2 good ones. First, gandanarimab, that's an amyloid-lowering drug. He said they decided to move it to phase 3, partly based on its pharmacodynamic effect, meaning it was clearly lowering amyloid.
Okay, so they saw the biological effect they wanted.
Yes, but it wasn't just that drug's data, it was also the wider evidence from the whole field, showing this link between lowering amyloid and seeing clinical benefit, often measured by something called the CDR sum of boxes.
So confidence came from the drug and the context of the field.
Exactly. Now contrast that with prasinumab. That's an anti-alpha-synuclein antibody for Parkinson's.
Different disease, different protein target.
Right. And in Parkinson's, Jeff pointed out, they don't really have those reliable pharmacodynamic biomarkers yet. They can't easily measure if the drug is hitting the alpha-synuclein target in the way they want.
So without that clear biological signal.
The phase 2 clinical data becomes much, much more important for deciding whether to go to phase 3. It really shows how vital those biomarkers are for making informed decisions, especially when you do have them.
Yeah, I can see that. Without proof the drug's hitting the target biologically, you need stronger proof it's actually helping people before you commit to a massive phase 3 trial. So what about completely new targets? Where there isn't all that prior data, how do you build confidence there?
That's a tough one. Jeff Cummings talked about this idea of triangulation.
Triangulation, like coming at it from different angles.
Pretty much. You look at evidence from human genetics, carefully used animal models, newer human models like organoids or cellular systems, and this is key, actual human tissue samples, like from brain banks.
So piecing together the evidence from lots of different places to build a case for the target.
Exactly. But, Jeff Cummings also gave a pretty sobering statistic. For a drug hitting a novel target, going into humans for the first time, the chance of success is still really low, maybe around 5%.
5%. That really drives home the risk involved in pushing into new territory.
It does, and Philip Scheltens, the investor, he echoed this need to look beyond just animal models. He said they often push companies to use human brain tissue in their preclinical work to really validate what they're seeing.
Trying to bridge that gap between the lab models and actual human biology.
Huh.
Okay, let's shift focus again to something equally vital, brain health as public policy. Miia Kivipelto is a huge figure here with her finger study.
Yeah, the finger study really showed lifestyle interventions can make a difference for cognitive health.
So Miia asked the big question, is the evidence strong enough now to make these finger-like programs part of public health policy?
And her answer was a clear yes. She feels there's enough evidence to really promote brain health across the whole lifespan. Starting young even, with this idea she called high five for life.
High five for life. I like that. So it's not just about preventing decline later, but actively building brain health throughout life.
That's the idea, and importantly, she stressed it's never too late. These lifestyle changes can still be beneficial even if someone already has a dementia diagnosis.
That's such an important message of hope.
It is, and she pointed to that Lancet Commission finding that something like 45% of dementia cases are linked to risk factors we can actually modify: lifestyle, vascular health, environment.
45%, that's huge. So the finger trial and the whole worldwide fingers network that spun off from it.
Exactly. They provide real evidence that these multi-component interventions work. Diet, exercise, cognitive training, managing vascular health, doing it all together.
So it's not just theory. There's solid trial data. Did she talk about getting policymakers on board?
Yeah, she mentioned the need for simple, clear messaging. Apparently, they had success in Sweden with new guidelines based on the finger model, partly because the message was straightforward.
Makes sense. Keep it simple.
And she also made the point that these interventions aren't just for brain health. They help prevent diabetes, stroke. It's a win-win-win.
True. But coordinating it must be tricky, right? Whose job is it to push for risk reduction across all these different areas?
That's a challenge she acknowledged. And she also said, look, research still needs to continue. We need to understand newer risk factors better, like hearing loss, vision problems, and figure out which specific interventions are most effective for whom.
So the future is likely.
Yeah.
What? A mix.
Probably. A combination of lifestyle approaches tailored to the individual, maybe alongside drugs eventually.
Okay, but how do these lifestyle things actually work?
Yeah.
Howard Fillit asked about the mechanisms. Is exercise just about blood flow?
Miia said it's definitely more complex than just vascular effects. The reason multi-domain programs like Finger work so well is they hit multiple targets, multiple mechanisms at once.
Right, not just one pathway.
And they're actually learning more by analyzing blood biomarkers from the Finger participants. They're looking at pathways related to vascular health, yes, but also things like BDNF levels.
Brain-derived neurotrophic factor, growth factor for brain cells.
Right. And synaptic plasticity, the brain's ability to adapt, plus inflammation, oxidative stress. It's likely different people benefit through different combinations of these mechanisms.
That idea of personalized prevention again. Very cool. She mentioned Finger 2.0 as well, adding metformin.
Yeah, they're doing a big trial combining the Finger lifestyle program with metformin, specifically for people at higher risk for diabetes or who are obese.
Metformin, that's a diabetes drug, but it's often talked about for anti-aging effects too, isn't it?
It is. It seems to have multiple effects. So they're looking at whether adding it helps, what the best dose is, when to give it. It's a really interesting study.
Yeah. Definitely one to watch. Now, Philip Scheltens had a point about studies, didn't he? About randomized controlled trials, RCTs.
Yeah, he questioned whether short-term trials, say 1 or 2 years, can really capture the impact of things people are exposed to over their whole lives, like decades of diet or exercise habits.
That's a fair point. Can a short snapshot really tell the whole story?
Miia acknowledged it's a limitation. You have to design the study carefully, be realistic about what you can measure. And she also said, look, RCTs aren't always possible or even ethical for everything. Sometimes you need to combine evidence from different kinds of studies.
Like you can't ethically do an RCT on smoking, right?
Exactly. You rely on observational data and other evidence.
Okay, let's talk supplements. Howard Fillit mentioned patients coming in with bags full of them. What did the panel say about nutritional supplements for brain health? Is there good evidence?
The overall vibe was definitely cautious. Miia brought up the WHO guidelines from 2019. Basically, they reviewed the evidence and found there wasn't enough proof to recommend specific supplements for preventing cognitive decline.
So, generally, no strong recommendation.
Generally, no. With maybe a couple of exceptions like vitamin D if you live somewhere with little sun, or vitamin B12 if you're actually deficient, but not for general prevention in most people.
It sounds like the thinking is shifting more towards precision nutrition, fixing specific needs rather than broad supplementation.
That seems to be the direction. Reisa Sperling mentioned that in their trials, like the A4 study, they record what supplements people take, but it doesn't exclude them from participating.
So they track it,
Yep.
but it's not a deciding factor.
Right. And Jeff Kirsch raised some good points about potential downsides of supplements, adverse events, products not containing what they claim,
Yeah, the quality control can be an issue.
and also the opportunity cost. People might rely on unproven supplements instead of doing things we know work, like diet and exercise.
Good point. Philip Scheltens mentioned supplement use is lower in the Netherlands.
Apparently so. Less common there.
Howard Phil did mention a resource, though, for people wanting reliable info.
Yes, the ADDF website, cognitivevitality.org. He said it provides evidence-based reports on supplements aimed at both the public and professionals. Sounds like a really useful resource to cut through the hype.
Definitely. Cognitivevitality.org. Good to know.
And he also mentioned looking for the USP verified mark in the US as some indication of product quality and testing.
Okay. So bottom line on supplements. Be cautious. Focus on proven lifestyle factors and check reliable sources if you're considering them.
Pretty much sums it up.
All right, let's get back to the drug development pipeline. A huge issue is how slow it can be, especially with Alzheimer's progressing differently in everyone. How can we speed things up? Jeff Kirsch kicked this off.
Yeah, Jeff really stressed the need for better biomarkers again, but specifically mechanism-agnostic biomarkers.
Mechanism agnostic, meaning
Meaning markers may be digital, maybe from blood or spinal fluid, that predict whether any drug is likely to work on a regulatory endpoint, regardless of how the drug works biologically, whether it hits amyloid or tau or inflammation.
I see. A universal predictor of clinical success. That would be incredibly valuable for making go-no-go decisions earlier.
Hugely valuable. He mentioned tau imaging is showing promise, but it's not quite there yet as that kind of predictor.
Reisa Sperling jumped in on this too, right? Talking about digital biomarkers.
Yes, she highlighted things like tracking daily learning curves using smartphone apps. Could that give us a faster read on synaptic function, maybe in phase 2?
Faster insights would be great. And she also pushed for moving past some of the older assessment scales.
Definitely. Getting away from things like the ADS-Cog, maybe even the CDR eventually, towards more modern, sensitive measures of brain function. Miia probably mentioned one of their trials isn't even using the MMSE anymore.
Progress. So better biomarkers, better outcome measures. What else?
Innovative trial designs were mentioned a lot. Adaptive trials that can change based on incoming data. Decentralized trials, maybe using some of these digital tools, and the need for public-private partnerships to drive all this.
Recruitment is always a bottleneck too, isn't it? Getting people into trials.
Huge bottleneck. Reisa thought blood-based biomarkers could be a game changer for pre-screening people much more easily. Maybe making decentralized trials where people don't always have to come to a clinic more feasible, though cost is still a factor there.
And using existing healthcare systems more effectively for recruitment.
That came up too, leveraging Medicare in the US or national health systems in Europe to identify and reach potential participants faster.
Okay, another potential angle the panel touched on, neurotrophic factors. Things like NGF and BDNF. Howard Fleece brought this up.
Yeah, the idea of using these factors to help brain cells survive. It's been around a while, but Howard noted it's been tricky to drug the receptors, like TPRA for NGF.
Difficult targets. But is anyone making progress?
He mentioned one company that has small molecules mimicking these factors going into phase 3, but again, the cost is enormous, especially for recruitment in these big trials. He stressed the need for financial partnerships.
Always comes back to funding, doesn't it?
Often does. And the panel discussed the biological complexity too, making sure you trigger the right signals inside the cell when you hit these receptors. Jeff Cummings also raised a broader point,
Which was?
focusing on shared mechanisms. Things like autophagy, the cell's cleanup system, or immunosenescence, the aging immune system, because often aging brains have multiple problems, multiple protein buildups, not just amyloid or tau. It's the biology of aging.
So targeting these more general aging processes.
Potentially. But again, you need biomarkers and trial design suited for those mechanisms, which is tough when you're not expecting immediate cognitive effects.
And Miia connected this back to lifestyle too.
She did, pointing out that exercise and nutrition might be protective, partly because they influence thousands of molecules, potentially dampening inflammation and supporting neuroprotection in complex ways. It's not just one simple effect.
And things like cognitive reserve, brain plasticity,
Hmm.
building resilience through activity.
Exactly. All part of the picture, even if they're harder to measure precisely than, say, amyloid levels.
Jeff Kirsch mentioned something called phase 0 studies. What are those?
Ah, yeah. These are very small, early studies, sometimes done even before the big toxicology testing. The idea is to check basic things like, does the drug actually get into the brain? Does it reach the target region? Does it seem to occupy the receptor?
So a quick, relatively cheap way to see if the drug is even getting to where it needs to go before you spend millions on tox studies.
Exactly. Jeff mentioned doing this with non-human primates even before animal tox studies, just to check the brain pharmacokinetics. Philip Scheltens agreed understanding biodistribution, brain entry, is a fundamental step you just can't skip.
Makes sense. Check the basics first. Okay, one last big topic. The distinction, or maybe tension, between disease modification and symptomatic treatments.
Right. Alistair Brown, joining from the UK, asked if the huge focus on disease-modifying drugs, like the antibodies, might be overshadowing the need for treatments that just help people feel better now.
That's a really important patient-centric question.
It is. And Jeff Kirsch's response was basically, for patients, feeling better today and having hope for the future are both critical. He feels there's actually been an underinvestment in purely symptomatic treatments, beyond just managing behavioral problems.
Why would that be? Is disease modification seen as more, I don't know, scientifically exciting?
Philip Scheltens kind of hinted at that. Maybe it seems sexier to investors, this idea of actually changing the disease course. But the need for treatments that improve daily function and quality of life right now is undeniable.
Reisa Sperling added nuance, though, right? Saying early intervention could change the pathophysiology.
Yeah, she argued that intervening early, even if we can't completely stop the disease, might still fundamentally alter its progression. But she also agreed that combination therapy is likely the future.
Combining different approaches, maybe disease-modifying drugs with symptomatic ones, or different disease-modifying drugs.
Both are possible, maybe using drugs in sequence or together. And Miia Kivipelto made the point that even the older symptomatic drugs, the cholinesterase inhibitors, might actually have some longer-term disease-modifying effects we're still figuring out.
So it's not necessarily either. We probably need both.
That seems to be the consensus, a spectrum of medicines for different stages and different needs.
Okay, so let's try and wrap this up for you, our listener. This deep dive, drawing on that expert panel, really paints a picture of a field that's, well, incredibly complex, but also incredibly dedicated.
Yeah, you see the scientific rigor, the persistence, from those really tough investment decisions Philip Scheltens talked about, to the potential of lifestyle changes Miia Kivipelto champions, the absolute necessity of better biomarkers that Reisa Sperling highlighted, and the push for faster, smarter trials Jeff Kirsch discussed. It's a dynamic landscape.
Absolutely. The big takeaways seem to be understanding the deep biology is paramount. Collaboration is essential. Academia, industry, funders, and the ultimate goal is always about finding real solutions for people affected by Alzheimer's at whatever stage they're at.
And a few things really stood out, didn't they? That 1% funding rate just, wow. It shows the mountain researchers have to climb. Also, how profoundly lifestyle seems to impact the brain through so many different pathways. It's not just one thing.
Right. And that ongoing discussion about balancing disease modification with immediate symptom relief. It really grounds the science in the reality of living with a condition. And of course, the potential for blood tests to maybe revolutionize screening and trials. That's a huge point of hope.
Definitely.
So we really encourage you to explore some of this further. Look up the finger trial, learn about multi-domain lifestyle interventions. Check out that website Howard Fillit mentioned, cognitivevitality.org, for solid info on supplements. And maybe think about the ethics around early biomarker testing as it becomes more common.
And maybe we can leave you with this final thought. Alzheimer's progresses slowly, differently for everyone. There's this growing focus on intervening really early, even before symptoms. So what's our responsibility as individuals, as a society, in making brain health a priority, not just late in life, but across the entire lifespan?
Yeah, something important to mull over. Thanks for joining us on The Deep Dive.